Hello, my name is Jonathan Castillo, and this is my Case Connection Unit 2 assignment video and script. I chose Case 1 and Case 2. I will go over Case 1 first. Case 1. So, pyruvic kinase is the last step in glycolysis. A phosphate is transferred from phosphoenolpyruvate to ADP to form ATP and pyruvate. Pyruvic kinase is regulated by the hormones insulin and glucagon. In general, insulin acts like an activator and glucagon an inhibitor. Insulin acts as an activator for pyruvic kinase by first binding to the receptor tyrosine kinase. This then forms a phosphorylated tyrosine PY residue that acts as a docking station for insulin receptor substrates to bind to. These attached insulin receptor substrates also form residues for signaling proteins to dock onto. Among these signaling proteins is PI3 kinase, which leads to the enzyme activation of protein phosphatases. Now, in the presence of increased insulin, these protein phosphatases activate phosphofructokinase 2, which phosphorylates fructose 6-phosphate into fructose 2,6 bisphosphate. Fructose 2 bisphosphate helps to activate the enzyme phosphofructose kinase 1, which phosphorylates fructose 6-phosphate into fructose 1,6 bisphosphate. And it's fructose 1,6 bisphosphate that is the primary activator of pyruvic kinase in most of the isoenzymes. For M2, L, and R pyruvic kinase isoenzymes, they are all allosterically activated by binding to fructose 1 bisphosphate. This promotes tetramerization and it forms tetramers out of the isoenzymes. But it is important to note, though, that the M1 isoenzyme does not bind to fructose 1,6 bisphosphate. This is because it already has a high constitutive activity and it's structurally different than the other isoenzymes. So, in comparison, glucagon primarily acts as an inhibitor for pyruvic kinase activity, as the binding of glucagon to a receptor will eventually increase cyclic AMP, thus phosphorylating and activating fructose 2,6 bisphosphatase. This dephosphorylates fructose 2,6 bisphosphate into fructose 6-phosphate, which is headed in the pathway of gluconeogenesis, not glycolysis. So other inhibitors to pyruvic kinase also include increased levels in ATP, which allosterically inhibits the L and M2 isoenzymes when energy is high. Phenylalanine and alanine are also inhibitors. Phenylalanine binds to the M1 and M2 isoenzymes, thus allosterically inhibiting and decreasing the affinity of pyruvic kinase for phosphoenolpyruvate. Alanine, though, is only able to allosterically inhibit the M2 isoenzyme, but not the M1 isoenzyme. Now, the preferential expression of pyruvic kinase type M2 over pyruvic kinase type M1 involved in cancer cells is mainly due to how the isoenzyme M2 can divert intermediates into another pathway that supports cancer cell development. The M2 isoenzyme is regulated by the binding of tyrosine phosphorylated peptide, which binds to the M2 isoenzyme, resulting in the release of fructose 1,6 bisphosphate and inactivation of the isoenzyme. This can allow for growth factor initiated signaling to glycolytic intermediates into other biosynthetic processes. These can be used for such processes like the pentose phosphate pathway. The NADPH and ribose 5 formed by the pentose phosphate pathway is used for nucleotide synthesis that directly supports cancer cell development. Thus, even though the M2 isoenzyme does not seem to be the ideal isoenzyme to be used in terms of glycolysis, cancer cells promote the expression of M2 isoenzymes in order to promote cell development. Case 2. When exercising, muscle glycogen is used in a process called glycogenolysis. What this process involves is converting stored glycogen to glucose 6-phosphate, which can be used for glycolysis and ultimately ATP production for muscle contraction. The specific enzyme that's used is phosphorylase kinase. So there are actually two different types of phosphorylase kinase. There's the phosphorylase A form, which is the active form, and there's phosphorylase B form, the inactive form. During exercise, the process of muscle glycogenolysis will specifically activate the phosphorylase A form, but in general, phosphorylase kinase activity is regulated by several activators and inactivators. Usually, this means shifting phosphorylase kinase into either the active or the inactive form. But when exercising, the active phosphorylase A form will be primarily used to provide energy during exercise. So there are three primary activators involved. The first one activator is epinephrine, which binds to a beta-adrenergic receptor on the muscle cell. This binding causes a conformational change that is transmitted to a G protein complex consisting of a gamma, beta, and alpha subunit, with a GDP attached to the complex. A GTP is switched for the GDP on the alpha subunit, which is then detached from the G protein complex. This alpha subunit and GTP then activates the enzyme adenylyl cyclase, which synthesizes cyclic AMP. This then leads to the second activator, which is cyclic AMP. So an increased concentration of cyclic AMP catalyzes phosphorylation via ATP to shift inactive phosphorylase B kinase to the active phosphorylase A form. The third then activator is calcium, which is used when binding of epinephrine. Also activates phospholipase C. Phospholipase C can cleave phosphatidyllinositol 4,5-bisphosphate, or PIP2. This cleavage forms inositol 1,4,5-trisphosphate, which releases calcium from the endoplasmic reticulum. This calcium then binds to the calcium sensor calmoludin on the phosphorylase kinase and activates the active phosphorylase A form. So overall, once glucose 1-phosphate is freed, another enzyme called phosphoglucomutase is used to isomerize glucose 1-phosphate into glucose 6-phosphate, thus finishing off the entire process. So now, taking long walks is still possible with a deficiency in phosphorylase kinase. Understand, though, that this condition is called McArdle disease or glycogen storage disease type 5. This condition is characterized by having a deficiency or complete absence of glycogen phosphorylase. Common symptoms of this disease do include pain and weakness in muscles when exercising, which are usually made worse when exercising. But this does not mean that a person can no longer do such activities as walking. It is possible to do moderate intensity aerobic training, which can increase muscle oxidative capacity over time. Studies have shown that this is possible due to something called a second wind phenomenon, in which taking a brief rest period after about 10 minutes of continuous exercise help reduce exercise-induced muscle pain. So it may be possible to take longer walks in time. Now, no, it is not necessary to eat outside of normal meal times, like in the middle of the night, to maintain blood glucose levels. First, it should be noted that the process of maintaining blood glucose levels is done through the liver only, which provides glucose to maintain blood glucose concentration. The process, though, is like what occurs in the muscles, as phosphorylase kinase A form is used in the liver. So a deficiency of phosphorylase kinase overall would reduce the amount of glucose within the blood glucose concentration. But it is possible that a carbohydrate-rich diet may help with blood glucose levels. This does not necessarily need to be done at a specific time of the day, but rather the focus is on the type of foods. Finally, excessive lactate will not occur during intensive anaerobic exercise with people that have phosphorylase deficiency. In fact, the amount of lactate produced is less than people with a deficiency. Though another reason why there is not an excess in lactate is because people with the deficiency tend to uptake lactate more than lactate is released in active muscle. This is because their bodies are using the lactate. So in the absence of being able to use phosphorylase, the enzyme lactate dehydrogenase is instead used to convert lactate into pyruvate for the TCA cycle. The bodies of people with a deficiency then are compensating by utilizing the energy produced by the lactate instead for anaerobic exercise. And here are my references, and they are also provided at the end of the script that I will be uploading. And that's all. Thank you.